Hi, my name is Sarah Portolano, and I'm going to be talking about the Piezo1 protein. So the Piezo1 gene encodes a mechanoreceptor ion channel protein. It's located on the lipid bilayer of the cell membrane as well as the endoplasmic reticulum membrane. And basically, the mutation of the Piezo1 gene is linked to DSH1 and xerocytosis. Its mutation can cause deformed erythrocyte membranes and changes to the intracellular ion concentration. For all of my models, I used air-dry clay in order to mold the shape, and I also used food coloring, and for two of my models, I used this Q-tip to represent the covalent bonds between the amino acids. For my main structure, Piezo1 has a trimer shape, meaning that it has like 3 identical subunits, and each of these domains come together towards the central cap. So this is supposed to represent, so here are the 3 blades, and this is supposed to represent the C extracellular domain, and then underneath is the ion channel, where mostly like potassium and calcium go through. So for the primary structure, I represented the non-mutated form of the amino acids, and so arginine is the correct one. And I also put like the mRNA on top that encodes for it, so it's GCG encoding for arginine. But for the mutated form, it's arginine's replaced by histamine, and GTG is encoding for it, so that just shows the mutation. For the secondary structure, I put alpha helices on to the blades to represent that the blades are composed of this folding pattern. And for the tertiary structure, these polypeptides, are folded up almost into a cup shape. So I tried to mimic that in this structure. And for the quaternary structure, this is the whole thing. It's basically, the 3 identical subunits, and that the ion channel's under, and then the CED is on top. So the mutation of the Piezo1 protein results in a gain of function phenotype. This basically means that there's an abnormal expression of delayed cation channel activations. And so the result is that the ion channel remains open for too long. The detailed information about how the mutation specifically affects the protein shape still remains under investigation, so it was kind of hard for me to represent that in my structure. But basically, I just kept in mind that the permeability of the ion channel is what's impacted by the mutation. So the missense mutation in the Piezo1 gene is linked to the increased permeability of the cations, leading to red blood cell dehydration. And due to this, red blood cells have an altered cell membrane shape and develop a characteristic central slit. So this is my red blood cell. And so the mutation of arginine to histamine is located at amino acid 2456. So this was my model. And the limitations of my model include that because there's, again, as I said earlier, little detailed information about how specifically the Piezo1 mutation affects the shape of the receptor, it's difficult to visually represent this. So that's why I mainly verbally explained it. And another limitation is that because I was making the entire mechanical receptor out of moldable clay and it had to dry, I just stuck to making a base structure and sticking the alpha helices to it. But obviously this isn't accurate, as the whole thing's composed of alpha helices. And my last limitation is I stuck the mRNA onto the amino acids in my primary structure to symbolize how the missense mutation occurred, but this is not accurate because obviously it's not on that.